Thank you very much, Javier, for agreeing to give a Build a Cell seminar, and you can get started.
Thanks so much, Kate, for the invitation. I hope you will enjoy my brief talk on what we have been doing in bottom-up approaches towards, so to say, very simple fibrillar networks that we try to replicate inside confinement. So we have two main research lines, which I don't know what to do with this. Yeah, maybe here. So which are focused on small molecule applications to biology or synthetic biology, or this field called the systems chemistry. In one hand, we like to prepare small molecules, small amplifiers or other molecules that we use to transport small ions through ion channels or pores across membranes, and then also to prepare synthetic amplifiers that we use to move bigger molecules or biomolecules inside cells, such as nucleic acids or proteins. This line is probably the one that we have mostly focused over the last, I would say, 7 years, and we have recently started also some work on this kind of. It's still early to call them cytoskeleton mimics, but functional fibrillar systems that we want to prepare with very simple synthetic molecules, so artificial monomers that we polymerize and then we study them inside vesicles or droplets or other systems. So I'll talk about today on this supramolecular in confinement, supramolecular polymers in confinement topic in my lab. I like to use this slide, this picture, to kind of share my vision or our vision, which is, I think, also shared by many of you on the emergence of life, which is a functional vision. So basically, we believe that, some of us think that maybe life emerged as a functional consequences of a given set of conditions in a particular environment. And so if you want to understand how these functions started or why they have started, it is very difficult to try to accomplish that or do that if you go from a top-down approach, because if you try to understand, for instance, how the transportation system works by looking at this top view image of a interstatal freeway connection picture, is going to make you confused. A lot of things you won't understand, such as why there are so many U-turns or all these signs, or why there are cars inside, many questions. But so if you try to address the question by asking the most simple functional question, why is a road made for, or why is a freeway made for, maybe you will have an easy task to replicate it under the starting conditions. So, for example, if you think about transportation systems, it won't be difficult to imagine at the beginning of the transportation system, for instance, at the beginning of the Roman Empire, and this is a small Roman road that you can find here in Galicia, plenty of them, and basically we used to say that all roads take you to Rome, right? Because at that time, basically they used these simple building blocks, such as rocks or plain rocks, to connect Rome with all the parts of the empire. And this is what we try to do in simple functions. For example, we are interested in the functions of simple early prebiotic or very early fibrillar systems that could have emerged in cells. We became interested in this topic because at the end of last century, not so far ago, it was discovered that in archaea there was a cytoskeleton. That discovery was shocking because it was believed that maybe cytoskeleton was a late in evolution organelle. The presence of these protofilaments in archaea was clearly demonstrating that this organelle was there from the very beginning. today is almost accepted that most, if not all, the bacterial and archaeal cells contain at least one protein filament. That is important because it means that probably all these one-dimensional supramolecular polymers could have played a role for making these initial cells better competitors, or giving them some sort of a functional advantage. And well, it's true that when we became interested in this field, there were already plenty of examples in the literature, but they were mainly using natural proteins, so mainly using natural building blocks. Here you can see one example of actin fibers that have been reconstructed inside a GUV, a giant vesicle, paper by Maeda. Here you can see a very nice reconstruction of moving motors in microtubules confined in a droplet, which are finally ending up by defining perfect tetrahedral nematic systems in this droplet, which is a nice example of how a very complicated natural system could give rise to a very simple physical behavior. But at that time, there was not so many examples of this kind of bottom-up reconstructions of fibrillar systems inside containers using fully synthetic building blocks. And so we started this project trying to apply self-assembling cyclic peptide nanotubes to get them assembled and fibrillated inside droplets, and then checking if they could have some functional roles or if we could apply them for something. This architecture are made of peptides, which are made of an even number of amino acids which have alternating chirality. They go LD, LD, LD. And in this particular configuration, they make the ring of the peptide form a planar structure, which is disposing the amide bonds perpendicular to the peptide plane. So in this configuration, they can stack on top of one another by hydrogen bonded networks. And then they offer a very nice and versatile supramolecular architecture, because it allows you to control the diameter of the nanotube that you are forming by simply adding or subtracting amino acids, and also to decorate the lateral decoration, the lateral surface of the tubes can be controlled by just changing the lateral side chains of the amino acids that you select. So we've played around in the past in soft materials with these and other applications, such as in this kind of hybrids of peptide nanotubes with carbon nanotubes that you see here. But when we studied the project, one of the first things we wanted to do is to replicate one of the most simple things of a cytoskeleton inside a living system or a cell. And this was just the simple one of the bundling. So not all the fibers or nanotubes that you can assemble in the nanoscale, they do follow this one-dimensional hierarchical ordering. So bundling is a common feature of fibrillar systems, right? Such as you can see here. This is a picture we took of actin in our lab, and it's always we always observe that actin, it's bundling at the cortex of cells or close to the nuclei, places where the actin needs to reinforce and sometimes needs to have a better transduction of the assembly of the nanoassembly into mechanical energy. They fibrillate together, they get together, and they keep the hierarchical assembly. So we wanted to get this out, right? To get this working. First, we wanted to have a peptide ring that would assemble in a one-dimensional supramolecular polymer and that will keep bundling in one dimension. So maybe this could be strong enough to, I don't know, do something to the container or something. And so for that, we designed this cyclic octapeptide, so 8 amino acids. This is basically an amphiphile. You can see here there is a hydrophilic region, and here is a hydrophobic region, which is flanked, both of them, by a histidine amino acids that has this imidazole group here that can be protonated and deprotonated close to physiological pH. So the building block was aimed to be protonated at acidic pH. So when those guys are positively charged, they will be repelling each other, and so the monomers will be soluble. When we would add a base, they will become deprotonated, and they will get together. They will get self-assembled, and they will get arranged in this kind of configuration with the pyrens all into the same sites to maximize the to hide them from the aqueous solvent. However, there is a mismatch here in the distance of the antiparallel beta sheet that is established between the peptide rings and the pi-pi interactions that are established between the pyren rings, which allows for a nice interdigitation of the pyrens, and this could keep the growth of the nanotubes in one-dimensional. And that was our first aim. We first measured CD, which reflects the sense of chirality. Here it's, for instance, this band is assigned to the pyren moiety. And you can see that the pyren is filling the chiral environment with the pH. And when we basify it from acidic pH to neutral pH, there is a emergence of this cotton effect here, that if we plot against the pH is giving us this steep curve, which is located at the pKa of the histidine, which is suggesting that we have a cooperative deprotonation self-assembly mechanism. At the nanoscale, everything looked fine. We have, by AFM and electron microscopy, nanotubes that have the diameter that would be assigned to one of these rings. And by rheology, we could confirm that we have some sort of a hydrogel similar to those soft hydrogels that can be prepared with natural building blocks or other soft materials. More interestingly, when we took a picture of this, we took some part of this gel and we took a picture of it, we could detect, we could observe in the microscope that we were obtaining not only nanofibers, but also not only nanotubes, but also micron length fibers here, as you can see in this image. Here, what we are using to image them is thioflavin. So we add a thioflavin there, which is a probe that rotates, and when the thioflavin is planarized in the antiparallel beta sheet, it gets bright, right? So then, well, we took these monomers and we put them in droplets, just water in oil emulsions, and then we trigger the formation of the fibers by adding base. Nothing very special, but well, we were just observing what was going on in there and if we could replicate this kind of fibrillation inside the droplets. Here is the epifluorescence measurements, and here is the confocal projections of the sample under acidic conditions. Everything is monomeric. So this is now we are checking the emission of the pyren, only emission of the pyren. And so it's blue emission. When we add the base, the pyren stacks on top of one another. And here what we can see is that the fibers turn green. This is happening because when pyrens get together, they stop emitting as a monomer and they have this excimer emergence, which is red shifted, green shifted, and so when they get together, they fibrillate like this. We could observe similar pictures like the ones that I showed you before of the actin by Maeda, and we could also detect that in the fibrillating droplets, we could observe many changes in the shape because the fibrils sometimes were like making the surface of these droplets more heterogeneous. We could also confirm that the droplets were transversal to the container, so they were crossing the whole interior of the system. Here, what we did is to add here the base propylamine, which is soluble in the oil, and it's diffusing through the system. And here we do the addition of the propylamine, and we could check how close to the addition of the addition point of the base we could get fibrillation, like in this gradient. And in most cases, yes, we could confirm statistically fibrillated droplets were changing shape more than non-fibrillated droplets. Well, that was one of the first things that we did, one of pioneers' papers on our group here. We just did this thing, was published in Nanoscale Horizons, and well, in this confocal reconstruction, you can confirm that the fibers are well distributed across the whole interior of the droplets. It's not the best confocal reconstruction. In the middle of this project, we got a new confocal microscope, so the pictures I'm going to show you later on are maybe more detailed. One of the things that the cytoskeleton is doing, which is particularly interesting for us, and I think for the functional purpose of this organelle, is to have control spatio-temporal distribution. So cytoskeleton is capable of doing these beautiful things here in the metaphase, microtubules organized in this way. Yeah, as I showed you before, this is the paper of Dojic, where the nematic phases are at the cortex of the droplets. And here is this nice paper by Sa, where they have asymmetric distribution of cytoskeleton fibers in GI and unilamellar vesicles. And this is another paper which I found some time ago, which is showing actin, I think it's actin fibrillation in the nuclei of cells, which is surprising, right? That actin can be, at almost everywhere, and it can, at certain times go to the nuclei and fibrillate there to help in, I think it was DNA repair or something like that. Well, what is interesting is that you can get actin fibrillated almost everywhere in the cell at a particular moment in time, right? So this is very interesting. So we thought, well, maybe we can try to replicate this system in this property in very simple models by changing the assembling triggers. So here is the monomer I showed you before, and there are two ways of triggering the assembly of this monomer. One is by protonation, but as I told you before, just adding base, right? To deprotonate here, they get together. But the other one could be adding salt. So if you add salt to a molecule that's ionized, if you increase ionic strength, maybe you can also shield the ionic repulsions, and you can get them together just without changing the base. However, the properties of the final compounds that we are getting will be different. So if we trigger the assembly by base, the will be deprotonated and very hydrophobic, or more hydrophobic, and when we trigger the assembly by base, because in this case we will get fibrillar bundles which will be very hydrophilic because they will still have the ionic character. So we thought maybe changing the chemical nature of the fibers by using different triggers of the assembly, we could control their location, because probably more hydrophobic tubes will tend to go to the cortex, where there is the water-in-oil interface, and very hydrophilic tubes under osmotic pressure might go to the close core of the droplets. And for doing this, we started by measuring this is, I think, fluorescence intensity. Here you can see the monomers, and this is this assembly here in yellow. And sorry, this is not, well, here is normalized, right? And basically, when we trigger the assembly by base here in red, you can see that the excimer part is higher, that the excimer emission is higher. So that means that the pines are in much closer contact. So probably this is also suggesting that the tubes that we are obtaining when we basify are more hydrophobic. And we could confirm the same behavior by UV spectroscopy. More quenching was observed when we basified rather than when we just were doing the charge screening, which was also quenching the emission, which was a good signal that we were getting assembly by adding salts to the system. So to have a good control over the salts, over the assembly triggers that we were using, we use microfluidics. So I think that you might be aware of this technique, probably even more than me. We use it here just as a tool to control the flow of the salt or the base that we were using, and basically we have a system where we are adding here in black is the oil, and here are the assembly triggers in pink, and here we are adding the peptide at acidic conditions. That's the video I used to explain the setup. Basically, we did this in collaboration with scientists in Cambridge. So basically, we put the peptide here under acidic conditions, and we just inject oil here, and then we generate the droplets in the outlet. And here we have peptide droplets that are coming from here, acidified. This is under acidic conditions. And then we do a pico injection of base here. We have these two electrodes to facilitate the fusion of the droplets. So, well, this is what we were doing, right? We were just adding different base at different concentrations of different salts to check if we would control the assembly and perhaps the positioning of the nanotubes that we were observing. Here, now you can see before the droplets that we were making with the emulsions were very heterogeneous. Here we have a homogeneous population of droplets under acidic conditions like blue emission and nothing there, nothing assembling there inside of these systems. This is the profiling. When we trigger the formation of the fibers by base, we could observe a ring around the droplets, which was confirming that we have here the fibrillation at the cortex. And happily, when we add for instance, here calcium chloride, we could confirm, maybe it's not easy to see here, but we could confirm that we have fibrillation more at the core of the droplets. Here, maybe we have these confocal projections. Here is the sodium hydroxide fibrillation. So hydrophobic tubes are going to the cortex. Here is calcium chloride. More hydrophilic tubes are going to the core of our droplets. As we could control the triggers of the assembly, we were just thinking maybe by controlling the flow rate of a salt, here the valent cation, using this divalent counterion, sulfate, sodium sulfate, we were controlling the flow rate of this salt, and the higher the flow rate we were putting here in the inlets, we were expecting to have a controlled distribution of the fibers, and this was exactly what happened. So if we had a shock, a strong shock of sodium sulfate, the fibers were concentrated at the core. If we were adding less concentrated solutions, we have more heterogeneous distribution of the fibers. So once we could control this, we thought maybe we could apply this for something. And one of the things we thought maybe we could control, now we can, have more or less bundling at the cortex of the fibers. Maybe we could use this to control the communication between fibers or the assembly of, the communication between droplets or the assembly of those droplets. And this is what we did. We tried to get droplets together by increasing the concentration. Here is the, when we use nothing, I mean, this is just a monomers in acidic conditions. So it's an heterogeneous distribution, heterogeneous aggregation of the droplets. They get together, they crumble like this. But when we did the assembly of droplets when the fibers are at the cortex, we got this massive aggregation and this massive bundling of droplets in this kind of, well, I would say heterogeneous amorphous kind of systems. However, when we did the assembly of droplets experiments using droplets that were fibrillating with salt, so meaning fibers were at the core of the droplets and that were smoothly diffusing to the cortex, what we obtained were these kind of perfectly organized hexagonal well-packed networks of droplets. So you can see here is like a very nice reproducible pattern. We could get them as far as we want, as big as we want over the microscope. And here, this is funny picture. Basically, when we were doing the experiments with the cortex droplets, so with base here, and we have not enough droplets to get them together, we obtained these pictures, which well, they look like it's a fake, right? It's just a funny picture. They look like a cytoskeleton, but I mean, basically, what we found is that in small droplets, when they collapse it, the fibrillar networks were like radiating outwards in all the dimensions of the droplet. But when the droplets were collapsing in one particular direction, the fibrillar networks were adopting were like elongated on those particular directions, which was interesting picture. So one of the things that remained for us was to try to get this. I mean, at the moment, we were just fibrillating droplets by changing the conditions, the exogenous conditions. So we have some monomer, we add a base or we add a salt and we trigger formation of fibers. But we wanted to do something more like cytoskeleton-like by using a chemical reaction. This is a complex network of reactions there. Phosphorylation, dephosphorylation triggers assembly and collapse of the tubes and infarct from equilibrium systems. But so at the beginning, we were just focusing, well, we might be interested into getting these fibrillar systems by just using a chemical reaction. That was the initial single aim of the project. And we thought about, in this case, using these tubes, which have been developed by Professor Samuel Stubb in Northwestern University. They are based on hydrophilic regions, so basically a couple of hydrophilic amino acids. Here is a couple of glutamics. We have been using glutamics. This is important. They are anionic. Then they have a beta sheet seeding region. So these amino acids will enhance the formation of beta sheets. And then they have this hydrophobic tail. Here, the number of carbon atoms of these aliphatic tails is between 14 or 16. These are long aliphatic tails. Those guys get like this assembled, so the hydrophilic head looks to the solvent, the hydrophobic tails looks to the interior. Basically, what we did at the beginning was just to use an oxide connection between the peptide part and the hydrophobic tail. So what we were aiming for this was just to find out the best combination of peptides and tails that will allow us to get a working chemical reaction that will again maintain the hierarchical assembly. As I told you, it's not always the case that you get a nanofiber that will get in a nice micron dimension tubular system. And so we wanted to get this nano 2 micron assembly at the suitable time scale for imaging them in the microscope. So basically, we made these things because we can make this alkoxamine, and these aldehydes, these aliphatic aldehydes, are all commercially available, and we can connect many of them, make libraries, and then find out where are the best combinations to get fibrillars at the micron size. Well, we screened some of them, and we found out that the good combination were always, in many cases, using tetradecanal, right? So a carbon tail of 14 carbon atoms, right? And so you can see here that with this one of these peptides with some anions with a couple of carboxylic acids and a tetradecanal tail, we were obtaining, well, it was a nano system, but the nano tubes were also forming fibrillar systems that we could image here, again, using the thioflavin probe I mentioned you before that assembles together and then forms the micron fibers. First problem we got here is that when we wanted to do this in situ in the water, tetradecanal was extremely insoluble. As you can imagine, right, 14 carbon atoms is not very water soluble. But it was not difficult to find a solution for that. We basically just cut the hydrophobic tail in two. So here what we have is that we have incorporated part of the tail into the peptide, which is still soluble because it's forming some small micelles, which are not giving any signal in DLS, but very small aggregates, so still very soluble. And this octanol now is more soluble in water. So we started analyzing the reaction of these two guys in water by HPLC. And what we found here is the disappearance of the peptide head that we have checked by HPLC. So we normally observe lag in time and then started decreasing of the head, so giving some sort of onset of the reaction after some sort of delay. So we thought maybe this could be some sort of physically autocatalyzed process, and we could observe that this was when we were measuring the circular dichroism, so emergence of the beta sheet region here, the chirality of the beta sheet, when we plot the exciton coupling against the time, we were observing this sigmoidal curve, which was supporting this kind of mechanism. And well, we suppose that maybe the connection of these two amplifiers were giving rise to aggregates that will improve the solubility of those reagents and will also bring them in close proximity, accelerating the formation of the product. We confirm this physically autocatalyzed process by adding seeds to the system. So basically, we took a bit of the final product of the reaction, the two of the reactants together, we add a 5 mol% to the reaction, and then after seeding, the lacking time for the disappearance of the head was gone, which was confirming the mechanism we were supposing. After that, we were expecting the formation of nanofibers and then nanobundles of these fibers, and this sadly happened in the microscope. In this case, we were observing, this is in the bulk water, we were observing the emergence, the abrupt emergence after some 5 times or so of this Eyster-like fibrillar system. We then put these monomers, the peptide monomers in droplets, and then we add the octanol from the exterior to check if the fibrillation process could happen across the interface of this quadrinol suspension. And we could confirm by fluorescence here in this case, again measuring the fluorescence of thioflavin, that we have this kind of sigmoidal curve with some lacking time that we could reduce by adding progressive amounts of the seed, and did some control experiments to confirm the amphiphilic nature of the process. When we measure the pictures, here is the addition tone of the octanol, we could check this gradient-dependent fibrillation of the droplets. But when we analyze a single droplet, then we observe the following. We could check that after some time, especially close to the addition of the aldehyde, we were observing an abrupt emergence of the fibrillar system that was distributed heterogeneously around in all the volume of the droplet. But with the time, it was accumulated at the cortex. That was an interesting experiment, and when we were checking at the videos of these fibrillating systems, we could find accumulation at the cortex, and then some sort of uptake phenomena that were happening, or sucking of the context, or more uptakes, like the bigger droplets were like taking up the smaller ones by this kind of enhanced fibrillation. We could quantify those kind of fibrillating at the cortex. It was statistically demonstrated that fibrillar networks trying to accumulate at the contacts of neighboring droplets. So we thought, well, maybe this fibrillar system is playing a role with the exogenous components of the droplets. So maybe we can try to check this reaction in the presence of different things, different things that we will add to the suspensions, and check if the fibrillating systems are triggering some sort of interaction with some of the exogenous components. For that, we just basically run the same process in the presence of several dyes that we have around in the lab. For instance, this is rhodamine B, sweet-side ionic amphiphilic fluorescent probe. This is cationic version of this probe. So basically, we just have an ester bond here, which brings the probe with a net positive charge, and then another positive charge dye. So in the presence of rhodamine B here, we could observe that yes, the fibrillar network was being formed. As you remember, it's anionic but amphiphilic, so it's normal that it will take up some amphiphilic dye. But beyond the uptake, nothing special was going on here. Here, however, with the rhodamine 6G, we could only observe not only the uptake of the probe and the enhanced fibrillation, but at the same time, a critically enhanced rate of the coalescence of the droplets. So that was suggesting that we could have some sort of a selective uptake that would be controlling the interaction of the bundles and the future coalescence of the droplet, which is easily explained because our anionic fibrillar system will be relaxed, the repulsion between fibrils will be relaxed with this cationic probe, which might enhance the fibrillation and the bundling and the subsequent collapsing and coalescence of droplets. Here is the experiment with HOH. This is a dye that we use to label DNA in cells, which is cationic, and again confirm not only the fibrillation, here is in green our fibrillar, and in blue the HOH, and you can see that they are formed together, and that sometimes, you know, droplets are even rotating, right, to maximize the fibrillar contacts, as you can see in those examples here. We could quantify those things after reviewing, but the take-home message is that we could, you know, do a fibrillation that actually the message is that you don't really need such a complex machinery to trigger a sophisticated behavior. So here, just the connection between two small molecules triggers the formation of an amplifier, which physically auto-catalyzes its own synthesis to trigger the bundling at the cortex, the uptake of exogenous substances, which at the end turn out to control some sort of coalescence or predatory behavior of those droplets. Final experiments that we did was to encapsulate a cascade enzymatic systems between droplets to check if fibrillation would make these two populations of droplets communicate. We put a glucose oxidase here and whole redis peroxidase in the other droplets, and then we encapsulate the substrate, the glucose for this enzyme. We encapsulate it in the other droplets. So we could, and then if this enzyme produces hydrogen peroxide, it will oxidize if the hydrogen peroxide is also exchanged. It will oxidize some dye here that will give us red fluorescence if there is an exchange of the components, right? So we were like expecting fibrillation will make these droplets to talk, to exchange components, and then we could monitor that by the emission of red color. And we could observe here, for instance, in this droplet, that was the case, and especially those droplets that were giving us highest fluorescence, they were giving also the highest content of fibrillar networks at the cortex of the droplets. That's all about the science. I wanted to tell you because maybe you are in the States and you are not familiarized with Santiago de Compostela. I'm coming from this place. This is Spain, right? This is here France, right? This is the end of Europe, right? Here is Santiago de Compostela. Here, this red star. Our city is kind of known because there is a pilgrim walk, right, which comes from the north of France and from the south of Portugal, because we have here this cathedral, which is where the St. Jacob bones are supposed to be buried, right? This is a nice picture probably took during the COVID-19, right? Because this is never like that. You know, this guy has been very lucky to be alone there at that time. Normally, this is opposite to the cathedral. This is this building, and this is the most familiar look of this Obradoiro Square that I remember. We have here plenty of cultural activities and also very nice seafood. Obviously, this is the most important slide. This is the people who did the work. I work with an impressive group of co-workers, and I would like to mention the names of those involved or partially involved in these projects. Projects related with cyclic peptides were mostly done by Alejandro Mendez Ardoy, who was a Marie Curie postdoc in my lab during that time, who's now assistant professor at the University of Sevilla. He was helped sometimes by Nacho Insua, who was postdoc in my lab, also Marie Curie in my lab, and now Ramón y Cajal in the University of Santiago. In the beginning of the project of the linear peptides, Kibon Bach also assisted the group in the synthetic experiments, and the fibrillar system was made by this guy who's just behind me here, who's this tall guy who's Richard Both, who got a PhD from the group of Stephen Maggie, who's now postdoc with Chris Keating in Penn State. I'm very thankful for all the funding that I have received, especially for this project, for the Human Frontier Science Program, and also the ERC, who's contributing. And I thank you very much, Kate, for giving me the chance to tell you a little bit about this research. Thanks a lot.
Thank you so much. That was a fascinating talk. There are some questions in chat. Can you see chat? If you could read the question before answering.
Go to the chat. Yeah, if I find it. Thanks for the comments. Does fibrillation depends on pH, and how is it affected by molecular candidate? Yeah, very good question. You know, this is a question that we also have. Yes. If in terms, it turns out that if you, it's a very important question. Both of them. You know, we don't have enough time to really look into it in detail. We would love, you know, to have more detailed studies, but we know that if we trigger the basification very strongly, right, this can affect the nanotube fibrils are kind of robust, right? They fibrillate well between pH like I don't know, between 8 and 10, they are fine. If you trigger the fibrillation like very strongly, like if you add a lot of sodium hydroxide, then fibrillars, the networks are not as smooth. They might get some sort of like a precipitate, they aggregate too much. So definitely yes, the pH, how do you basify the system affects a lot the fibrillar, the shape and the chemical nature of the fibers that you are forming. And in fact, yeah, if you go too hard, then they precipitate and it's not nice. And then molecular crowding, this, yes, I mean, we didn't try, but we know it will be yes, right, for sure, right, because we know that when we change the ionic strength, fibrillar system changes also, not only but we know also the rheological properties of the solutions also, they change. Right, but yeah, definitely, right. The molecular crowding will influence, and we are applying to some cost actions in the past to try to get more expert co-workers in molecular crowding to try to look into this more in detail, right? Because this is not our area of expertise, and we did not go into details, into experimental detail, but we know it will be yes, it will be a yes, and we will be very interested in to study how these systems behave, synthetic systems behave in crowded environments. But we didn't have time to study them, right? But thank you for the question. It's very, very, very good. So another question. Yeah, I see the question from Tanner, right? But I don't understand very well. So to make it inside a synthetic cell, like you want like to use to fully use? Yeah. All right. Yeah. So in fact, we are taking the steps in this direction as well. Good question. And for instance, what we are doing is that we are using, because one of the things that we need now is to increase. If we want to get more functional behavior, we need to increase the amount of the molecules that we are using, right? Because the solubility of some of the reagents we're using is very low. So one of the things that we have used to increase the concentration of those systems is to use enzymes to generate the compounds that we want. For instance, oxidases, right, to generate aldehydes, which makes alcohols more soluble. We are having a lot of good results on those systems. You know, I still cannot tell you a lot of details about it, but this project is still alive and breathing. Yeah, I would say, and we would be. I mean, it's highly advanced at the moment. We are looking forward to more experiments on these things to complete some of the works we are trying. Yeah, Tanner, thank you for the questions. Yes. Yeah, probably there could be one of the other ideas that I always have is to disconnect them in more pieces, right? To make them in more small pieces, which will allow like for networks of enzymes, right? To do one reaction at a time. And this also will enhance a lot the solubility of the system, right? Which is a good property. Petra, those assemblies. Yeah. that's the problem, right? So when you generate such amplifiers in the presence of vesicles, you sometimes destroy them, right? So we are not so cool with vesicles, right? So we tried a bit with Neil Devaraj, right? And at the beginning, we did some experiments in GUVs, and the system was not so very stable. But yeah, we will be looking forward trying those in vesicles. And yes, of course, it will be very nice to do them in vesicles because yes, in vesicles you could do more sophisticated systems and things like that. We might be needing more robust lipids or less amphiphilic fibrillar systems or less concentrated fibrillar systems. These are projects that will require optimization. This will take time. And yeah, you know, people, it's always in a hurry. Can you theoretically model those? Oh, yeah, we can. We can model them. We have a friend, Rebecca Garcia Pandino, who's helping us in modeling them. But what for, right? I mean, Angela Sarik, right, is doing so much work on this thing. Yeah, she's doing. We are not computational, but for sure, right? As you model proteins, those are simpler than proteins, yeah. And of course, yeah, for sure, it will help predict assemblies. But we sometimes do some work with Rebecca Garcia Pandino, our expert in computational chemistry, associate professor here close to my desk. Yeah, of course we can model them, yeah. And yeah, they could be used to predict, of course. And not only to predict them, but the coarse grain. We have some projects now that allow us to predict assemblies of these fibrils across different dimensions, right? How they can get together not only as a fiber, as a one-dimensional, but there's 2- and 3-dimensionals. Yeah, very interesting. Yeah, this work is ongoing. Sure. Inside a liposome with all free protein expression system, doable. Yes. Might take time. Yeah, optimization time. We are in the way to that. I mean, yes, of course. And very likely when you form the fibrillar systems, those will affect the GUVs. Yes, probably divide them, most likely. Could you introduce modifications such as yes, of course. We could modify especially with the cyclic peptides, we could modify them. I'm answering here Luis Daniel. Yes, we could assemble them and we could put them like I don't know, clickable units in the side chains of the peptides, and then we could click them anything we want. And yeah, once I mean this is another thing that we will also like to study how connection between this is a lot of work in natural proteins, right? How they get together, right, with all these helper proteins that get them. Yeah, we could use very simple systems, right, to connect networks and everything. And yeah, another thing that maybe nice to do. Hey, Andreas, how are you, man? Nice to see you. How are you doing? You have a lot of figures. Yes. Have you? In fact, the new work we are doing is with very good question. Andreas Walter, my friend. I gotta get you here to Santiago, Andreas, my friend. This year we cannot wait more. Yes, the new work we are doing with enzymes and everything is in dextrin pegylated systems. The thing is that it's very nice because some of the functional consequences of the system changes, which is interesting, right? And of course, surface tension play a role there. Of course, and we get those effects of crowding effects. Nice to see you, man. I'm missing you.
My pleasure. Nice to talk. Thanks.
Now I'm going with Nukt Avert. Thank you, Professor. Yes, as it's like the same as Andreas. Coacervates also interesting systems. Obviously, yes, and well, I mean, we would be very, very interested in any collaboration with anyone who had a robust vesicular system or who has a coacervate system to send our amphifiles and to let them know how to get them assembled, right? Because it's very easy for us to prepare them, right? And of course, we are not so expert in UV. We try to make them by different systems. I think one of the most promising system, we tried several things. One of the most promising experiments that we did was like this inverse emulsion, right, with droplets and with centrifuge, and then those vesicles were more stable, but still kind of tricky system. And we are not experts, so we give those more complex systems more aside. But if anyone is an expert on these things, we are more, and if he's interested in trying our amphifiles, we are more than happy to test them. Gabriel Lopez Naos says, beautiful work. Can you use them better assemblies to form pores between water and air droplets? Yes, we do think that we are forming pores. In fact, if you check those images, we have encapsulated one I mean, one of the enzyme is labeled with a probe. So one of the droplets is blue. The other one is with the whole range peroxidase is gray, right? And so if you can see, they turn red before you can see the micron fibers. But that is because the nano system is already working, right? Once you get the nano assemblies, and we could confirm by TM microscopy that at very short times what you have instead of micron is nanofibers. So at nanofibers they will be making pores, and in fact the pores are site selective, because as you see the blue color is not passing, so only the red color is passing, right? So that means that bigger molecules, the bigger proteins cannot cross things, but the small molecules, the amplest red that is being produced is being crossed, but not the bigger molecules, not the bigger proteins, right? So yeah, absolutely, pores are there, or transient dynamic pores, whatever the I don't know, maybe they are not like assembled barrels, right, or stuff like that, but dynamic transient pores are being formed there, and those are site selective because they don't allow the passage of the enzyme. Yeah, I think it's all.
I think you went through all the questions. Thank you so much. Yeah, I would be really curious to see how it works in vesicles, but I can see how that's going to be really hard.
Yeah, probably yes. Once you get there, you can try that. I mean, the best way would be to first prepare the vesicles with the peptide with a small tail, right? Then you add the other part. Once the amplifier is being formed, then things will happen. Whatever happens, I don't know. But the things will happen, that's for sure.
Yeah, totally. All right, if there's no more questions, I just want to say thank you so much. Thank you so much, and thanks everyone else. And see you all next week.
Yeah, Kate, if you don't mind, maybe we can have some small discussion. I wanted to.
Yes.
Yeah, all right.
I just chatted you. I'll send you a link.
All right. Thank you.
All right, thanks.
I'll go there.
Bye.
Bye-bye.